All right, let's get right into it. Today, we are unpacking gene mutations, those tiny, tiny changes in our DNA that can literally change everything. We're talking about the very engine of evolution, and sometimes the source of disease. It all starts with a single little alteration in the code of life. And hey, if you've got an honors bio exam on the horizon, this is definitely going to be the review you need.
So here's what we're going to cover. First, we'll lock down exactly what a gene mutation is. Then we'll get into the real nitty-gritty, the difference between a small point mutation and a massive frame shift. After that, we'll see what all this means for the proteins our bodies are trying to build. We'll also tackle a big question: are mutations always a bad thing? And to wrap it all up, we'll look at how our genes can be controlled without the code ever changing at all. It's going to be good.
Okay, first things first. Before we can really get into the different kinds of mutations and the chaos they can cause, we have to be perfectly clear on what we're even talking about. What is a gene mutation?
The simplest way to think about it is this. Your DNA is the master instruction manual for building, well, you. And a gene is just one specific recipe in that massive manual. So a gene mutation is just a change in that recipe. A typo, a different ingredient, a swapped word, any change at all to those coded instructions.
Okay, so we know a mutation is a change in the recipe. But now we've got to ask, how can that recipe get changed? Turns out there are two main ways, and telling them apart is absolutely critical. It's really the difference between a tiny typo and scrambling a whole paragraph. On one hand, you've got point mutations. Think of these like a single-letter typo. Just one little base, one point, is affected. But on the other hand, you have frame shift mutations, and these are a whole different ballgame. They don't just change one letter, they completely mess up how the rest of the sentence is even read.
So let's zoom in on those point mutations for a second. It seems like a small thing, right? One little change, but don't let that fool you. Altering just one single letter in that DNA sequence can, in some cases, have absolutely massive consequences.
Point mutations really come in three main flavors. First, there's substitution. That's a simple swap, like a C gets switched out for a T. Easy enough. Then you have deletion, where a base gets removed entirely, and insertion, or addition, where an extra one gets squeezed in. Now, pay really close attention to deletion and insertion, because even though they're just changing one single point, we're about to see how they can cause a much, much bigger problem.
And that brings us to frame shift mutations. See, your cells read DNA in groups of 3 letters, like 3-letter words. We call them codons. What happens in a frame shift, which, remember, is caused by an insertion or deletion, is that the whole grouping gets thrown off. From the point of that one little change, every single 3-letter word afterwards is now wrong. The entire message gets scrambled.
And just look at this. It shows you exactly how devastating that can be. The original code reads perfectly, making the amino acids methionine, glutamine, and lysine. But then, poof, one G base gets deleted, that's it. But because of that, the whole reading frame shifts over by 1. The cell now reads total gibberish, and it ends up making methionine, valine, and histidine instead. Every single amino acid from that mutation onward is wrong, and that is why frameshift mutations almost always result in a completely useless protein.
Okay, so we've got these typos in the DNA code. But so what? Why does it really matter? Well, this next part is all about connecting those dots, linking the change in the instructions to the final product, the proteins that actually do all the work in our bodies.
This really gets to the core of biology, doesn't it? That central dogma. DNA makes RNA, and RNA makes protein. So it's pretty simple. If you mess up the original blueprint, the DNA, you are absolutely going to mess up the final product. You might end up with a protein that's folded all wrong and totally defective, or the cell might not even be able to make the protein at all. This isn't just a theory. It is the fundamental cause of so many genetic diseases.
So the ripple effects are huge. Whether it's a completely broken protein, one that just doesn't work right, or no protein being made whatsoever, the end result can be an organism with a serious problem. And that's exactly how many genetic diseases show up. So far, I know this all sounds pretty terrible, right? Like mutations are just bad news. But is that the whole story? Not even close. Let's pivot now and look at the much more complete and honestly more interesting picture of the role mutations play. So let's just ask it straight up. When we hear the word mutation, our brains usually jump straight to disease or defect. But is that always the case? And the answer, you might have guessed, is a huge no.
See, whether a mutation is good or bad all boils down to its effect on an organism's fitness, which is just its ability to survive and have kids. If the mutation makes that harder, yeah, it's bad. But, and this is a huge but, mutations are also the ultimate source of all genetic variation. Every new trait, every adaptation, it starts with a mutation. Without them, evolution would just grind to a halt.
And for a perfect example of this, look no further than the rock pocket mouse. There's a mutation that gives some of these mice dark fur. Now, if you're a dark mouse on light-colored sand, you're an easy snack for a hawk. That's a harmful mutation. But take that same mouse with that same mutation and put it on a landscape of dark volcanic rock, now it's perfectly camouflaged. It's a beneficial mutation. It's all about the environment.
All right, for our last section, we're going to get into an idea that is just as powerful as mutation, maybe even more so. We're talking about the ability for life to change its traits without changing the DNA sequence one single bit.
So, think about that for a second. Is it actually possible to get totally different results, different traits, from the exact same set of genetic instructions? The answer is 1000% yes, and it all happens through something called gene expression.
Gene expression is all about control. Every single cell in your body has the exact same library of DNA, right? But a nerve cell is obviously very different from a muscle cell. Why? Because different sets of genes are turned on or off in each one. The genes that aren't needed get blocked or silenced, and this allows for all this incredible specialization from one single blueprint. And here's the mind-blowing part. The environment can be the one flipping these switches. Check out the Himalayan rabbit. The gene for dark fur is sensitive to temperature. It only gets switched on in the colder parts of its body. That's why its ears, nose, and feet are black. Or even more wild, look at these agouti mice. The mother's diet and nothing else can silence a gene in her babies, completely changing their fur color and how healthy they are. It's incredible.
So how in the world does a diet flip a genetic switch? It comes down to these little molecules called methyl groups. Nutrients in our food, like B vitamins, get converted into these methyl groups. And they can act like little sticky notes or tags that literally attach to a gene and tell the cell, ignore this one. They silence it, turning it off.
So when you put it all together, it's like our genes have this whole complex control panel. Some of the switches are internal, like hormones carrying messages around our body. But a lot of them are external, things like temperature. And as we just saw, even the specific food an animal eats can reach in and physically change which genes are being used.
Let's just end on this image, because it's so powerful. I want to say this one more time. These two mice have the exact same DNA. They are genetic clones. The only difference is that the mom of the small, healthy brown mouse ate a diet that provided those methyl tags, which turned off the agouti gene in her baby. It's just a stunning, perfect example of how our experiences can shape our biology.
And that leaves us with one last big idea to think about. We've seen that our environment, from the temperature outside to the food we put in our mouths, can fundamentally change how our genes work without ever touching the code itself. So if our DNA isn't some fixed destiny, but more of a dynamic script that our environment helps direct, what does that really mean for that classic debate of nature versus nurture? It really seems like the answer isn't one or the other. It's a beautiful, complicated dance between the two.